All right, so now we're going to talk a little bit about protein technologies and recombinant DNA technologies, and then at the end, just the sort of classic and reverse genetics, how we can go from a phenotype to a gene and how we can go from a gene to a phenotype. So as this figure shows, nowadays, using recombinant DNA technology, if we know a protein and we want to get back to its gene, there are techniques that allow us to do that. If we have a gene and we want to understand its protein product, we can do that as well. And this is all a fairly new, well, new to somebody my age, old to somebody your age. This is all fairly new in the scheme of things. Techniques nowadays, if we have a protein, you can get a little bit of amino acid sequence from it, use that to generate a DNA probe based on the genetic code, and then go fishing for the gene in libraries. If you have a gene and you want to understand its protein product, you can introduce it into E. coli, bacteria, or other simple organisms that grow rapidly, allow them to express it, and then study the protein. So nowadays, we can go from the protein to the gene and also from the gene to the protein, and that has obviously made it a lot easier for us to learn a lot about biological molecules without excruciating attempts to purify from complicated eukaryotic cells. Okay. So we'll talk a little bit about protein, and those of you that took Bio 311 probably will recognize these next 10 or so slides. So proteins can be studied initially by attempting to isolate them or at least partially purify them, stepwise separating of a protein from all of the other proteins inside the cell so that you can enrich for the protein that you're interested in and then study its properties or its regulation or its localization in more detail. So proteins can be separated from other proteins inside cells by various properties that all proteins have, such as their size or their charge, or their affinity or specific interaction with other molecules inside cells. And the most common mechanism of separation is something that's known as chromatography or simply columns. This is a vertical tube. It can be actually a standard pipette, plastic pipette, a pasture pipette, or just a long cylinder that you can buy, and it comes usually with a stopcock on the bottom so that you can open or close it, and some sort of plug on the bottom, and then you generate a column from some resin beads, and then you apply your sample, which is typically a whole cell homogenate. In other words, you take cells and you burst them open by a variety of means. Sometimes you just put them in a hypotonic solution and they take up water and swell, and then you can vortex or add a little bit of detergent, will dissolve the membranes, and the insides of the cell come out. You make a homogenate, kind of like putting something in a blender. And then your homogenate contains all of the proteins inside the cell, and then you apply them to the column, you let gravity do its thing, and then you wash through the column, and some things will bind to the column, others will elute or flow through, and then elute, and then you can enrich for the protein that you're interested in. Okay, so a little bit more of what that means. Okay. So one common kind of column is known as gel filtration or a size exclusion. In that column, you have beads, and the beads have holes in them. They look like wiffle balls, okay? They have, the beads are a uniform size, and the holes in the beads are uniform for a particular kind of bead, but you can buy beads that have tiny holes and beads that have huge holes and beads that have in-between holes. So the column is made of these beads with holes in them, and then you apply your protein homogenate to the top of the column. Proteins that are too big to fit into the holes in the beads will go between the beads and make their way through the column very quickly, so they are excluded from the beads or excluded from the columns as a function of their size. That's why it's called size exclusion. Proteins that are small enough to fit into the holes in the beads will get trapped in the beads. Eventually they'll find another hole and get back out of the bead, but that hole, then they'll fall into the hole in the next bead and they'll have to find their way out of that one. So their mobility through the column is slowed up by the fact that they are small enough to keep falling into the holes in each of the beads. So proteins go through the column as a function of their size. The proteins that are very large and excluded from the pores or holes in the beads go through the column quickly, as you can see here, and before you know it, they're coming out the other end. Whereas the little guys, the ones that are small enough to fall through the pores in the beads, get stuck in the beads and in between beads, and their mobility through the column is slowed down, and so they come out last. So you can use gel filtration or size exclusion chromatography to separate your protein from proteins that are bigger than it or proteins that are smaller than it, or both, okay, by using a combination of columns. And there's a variety of schemes that scientists typically use to get rid of everything bigger than their protein of interest by trapping it in the beads. I mean, eventually it comes through, but it takes longer. And then separating it from everything smaller, and then you just have a collection of proteins that are roughly the same size as your protein of interest. Okay. Another way you can separate, sorry, I was just trying to move that. Stop doing that. Okay, here, click and drag. There we go. Is to separate by charge using something that's called ion exchange chromatography, because if you make your protein an ion, it will bind to oppositely charged ions, and then if you wash with a high ionic strength solution like a salt, the ions in the salt will displace your protein and it exchanges or comes off the column. So you can buy beads that are coated with positively charged functional groups or negatively charged functional groups. The positively charged one, the common one, is called DEAE cellulose, and the A stands for amino. And the most common negatively charged ones are called carboxymethylcellulose or phosphocellulose. You guys know that carboxy groups and phospho groups are all negatively charged. So every protein has an inherent charge to it. It's the sum of all the positively and negatively charged amino acids that exist in a protein. And the charge of the protein itself can depend on the pH of the solution that the protein is dissolved in. So you can make your protein be uncharged, have a positive charge, or have a negative charge, and then your protein will either bind to the column or not bind to the column, depending on whether or not there's an ionic attraction. So to just develop that idea a little bit more, every protein has an isoelectric point, and that's the pH at which that protein has no net charge. So some proteins are rich in positively charged amino acids, some proteins are rich in negatively charged amino acids, some proteins have about the same. Okay? So every protein is going to have a pH at which their protein has no charge, because depending on the pH of the solution, you can determine whether or not the amino and the carboxy groups are ionized, right? at a lower pH, when there's lots of protons around, the amino groups will be positively charged and the carboxy groups will be uncharged. At a higher pH, where there's very few protons around, the carboxy groups will be ionized, so there'll be more negative charge. So by adjusting the pH, you can make your protein of interest have a charge or be uncharged. So what you can do, for example, is have your protein of interest loaded onto the column at a pH where your protein has a negative charge. It will bind to the positively charged beads. And then you can elute or get your protein off the column by washing with increasing concentrations of salt, which the salt ions will exchange for the protein, or by changing the pH of the elution buffer so that your protein loses its charge. If it loses its charge, it loses its affinity for the residue. Okay, so here's a picture of that as an example of what ion exchange chromatography looks like. And you have 3 different proteins in this mix, you know, the star, the little circle, and the big circle. So some of the proteins are not going to stick to the column at all. Okay, so they go in the top and come out the bottom. So this star-shaped one did not stick to the column at all. Of the ones that stuck to the column, in this case, they are being eluted by a salt gradient, increasing concentrations of salt. So the more tightly the protein is bound to the beads, the more salt or ions it's going to take to displace it. So in this case, this one is weakly bound to the column, and this one has a higher affinity for the column because it took more salt to bump it off. Okay. And the third kind of column, which is the most fun to do, okay, but is the most expensive, is to use something that's known as affinity chromatography. Affinity chromatography means the beads have something attached to them that your protein of interest binds to in a very robust and specific way. So basically, very little else sticks to the column except what you're interested in, and then you can elute it using anything that disrupts that interaction, salt or changing the pH. So in these cases, the beads, for example, if it's an enzyme, the beads might be coated with the substrate. The beads can be coated with, say, RNA or DNA if you're trying to pull out RNA binding proteins or DNA binding proteins. The beads can be coated with antibodies if you happen to have an antibody that's specific to your protein. So this is the most direct and successful way of specifically purifying your protein, but it only works if there's a bead available that has something that has a biological affinity for your molecule of interest. Okay. So once you have fractionated your lysate and you want to start looking at the proteins, you can look at them on gels, polyacrylamide gels. This forms a matrix that proteins will run through as a function of their size, because first they are totally denatured by heating them in the presence of a detergent molecule known as SDS. So that the three-dimensional structure is undone by using something that breaks the disulfide bonds, and by heating them in the presence of SDS, they get coated with SDS, which is negatively charged, so they'll all run towards the positive electrode, regardless of the natural charge of the protein, because by treating them with SDS, you give them a negative charge. So they all run as a function of their denatured size towards the positive pole. and you can view the entire collection of proteins by staining with dyes that just visualize all proteins, or you can look for your protein of interest if you have an antibody that's specific to it, because the antibody will bind to the protein, and then you can use a variety of techniques to detect the antibodies. These are referred to as immunoblots because you're using immuno or an antibody to detect your protein of interest.